Science Institute, the GSI, and I'd like to welcome you all to this fall seminar series where we have a combination of technology talks as well as faculty academic talks. We try to balance the number of academic talks with technology talks so that we don't have too much of the either. And for any of you students, postdocs, faculty that are interested in inviting faculty speakers to the GSI seminar series, please reach out to us at gsi@bu.edu. One of my colleagues, co-directors, Dr. Alec Grishaw, can help organize with you, pick a good faculty member to invite, or with technologies as well. Like today we have Singular Genomics that Yuri Alexiev, the sequencing core director, will tell you more about. We can also host those as well. So there's food I think is getting set up. Do you want to do the food after the seminar?
Yes.
Yes, and so that way we can get the seminar going. Yuri, can you introduce the speaker?
Hi everybody. Pretty good turnout for a technology seminar, I would say. Thanks for coming, you all. So today we're having Singular Genomics. As you know, next generation sequencing is the technology that I'm not afraid of this word revolutionized the way we do research nowadays. And from being on the forefront of the progress, it's now became sort of routine technique without which we cannot do any research. And the company named Illumina was enabling progress in this area for a long time. And they were actually eventually monopolizing the whole field, and progress sort of slowed down, as did the cost efficiency. But eventually their patents with which they protected their technology expired, or some of them expired, and that allowed a new competition to begin. And Singular Genomics was one of the first players who made next generation sequencing exciting and interesting again. They managed to create a competitive instrument from ground up without any reverse engineering or anything like that, which allows you to do cost-efficient next-generation sequencing. On top of that, it gets even more interesting because it turns out that this instrument actually can be upgraded to a spatial instrument, which allows you to do targeted transcriptomics, targeted proteomics, and even in situ sequencing for variable regions for which it's hard to make probes. And so we are leasing this instrument, not the spatial upgrade, but the general sequencing instrument. So we hope that you guys will eventually use it, and then we can think about if we are interested in upgrading it to the spatial instrument. So it's my pleasure to introduce Jonathan Slashinsky and Jordan Williams. Jordan Williams has a PhD from University of Southern California, and now he is a scientist at Singular. And Jonathan is the head of sales of this company.
Just the sales guy, not the science guy, even though I do have a BS in biology. But.
Well, I deal a lot with salespeople, so it's important. Yeah, so welcome. Thanks for coming here, and we hope for exciting.
Hey guys, the lunch, sorry, it came a little late. It is set up in the back. We still have we made a little miscommunication. I just need to put my slides on her slides. Veggie and salads are to the right, and then there's turkey, chicken, and BLT sandwiches. But let's go get sandwiches first, and then we'll come back in about 10 minutes. We'll start this talk. I don't want you guys to have to wait for lunch. Veggie and salads are on the right, and then the Bravo is turkey, there's chicken, there's BLT. Now, we got that.
Check quickly about the pilots and
Hey, you were so awesome, by the way.
Of course.
Seriously, like, thank you so much for all that. It was really, it's just nice to know that we have support like that.
Yes.
Wow, that's cool.
You guys came from Cali.
Were you from Boston?
Yeah, I'm from Boston.
Well, girl.
Yeah, I've been here for a while.
That's it. I'm from New York. And I'm California for like 8 years.
Nice.
Manhattan.
Oh yeah.
So I'm going to come out here.
No, you're fine. I'm always going to Manhattan. It's so fun.
So it was just so funny, like, just better than I was getting so stressed out.
I
Food delivery. I know, see, this is exactly how it was set up. So. Yeah.
Yeah, I was there before. So I was part of the spatial project with my own. So they were competent singular about spatial collaboration, not just
Yeah, we had all the samples we can run right now. But I just want to make sure that we can isolate either self-engine or triangle. If we can do it, then I want to make sure that we have that sample.
we just want everybody to get to the party.
Oh, and then I'm gonna give you the microphone.
Yeah, I'm pretty sure because it's
They want it for them.
and I'm reaching out to find
Okay, can you guys hear me? Yeah?
Hi.
All right. Since that was really easy, you guys got your food so fast. So, as Yuri said, my name is Jordan. I lead our field application scientist team at Singular Genomics. What that means is our team goes out and trains our customers. We pass along technology information. We help our customers apply our technology to their research questions. So, happy to be here. Happy to talk to you guys about the G4 sequencer. I'm realizing now that this is a different kind of seminar than I expected. So let me first get a hands of who all is familiar with next generation sequencing. Like, do we need to go through, you guys are all familiar with next gen sequencing? Do we need to go through what next gen sequencing is? Hands? No? We feel good? I'm gonna give a light summary of what next generation sequencing is. So next generation sequencing, what it does, as you already said, it revolutionized how we're doing kind of research in that it allows for the sequencing of multi-million DNA sequences all at once. So different chemistries, as Yuri mentioned, Illumina, there's a ton of sequencing companies out there right now that sequence in a different way. Singular is similar to Illumina in that we do sequencing by synthesis, and what that means is that we basically do a PCR reaction, so we're amplifying DNA, but instead of incorporating kind of dead nucleotides, I don't want to say dead, but non-fluorescent nucleotides, which is like a normal PCR, we're incorporating fluorescent nucleotides. In our chemistry, we use 4-color chemistry, meaning each nucleotide has its own color. We built, as Yuri said, all of our technology from the ground up. We didn't have to wait for Illumina's patents to expire because we created our own patents. So that's the first thing. So just imagine a PCR reaction where a nucleotide's getting incorporated in. It has a color. We take a picture at that cycle, then we cut off the color, we cut off the stop, and then we add another nucleotide, take a picture of that color, cut it off. And we do that very quickly in order to sequence your DNA strands, many millions of DNA strands. Okay, any questions on that? Okay, great. So for this talk, I feel like my head is just going to be in the screen no matter what. Maybe if I, like, does this thing work?
It doesn't work.
Okay. Huh?
It's a receiver end up. You
I have no.
got this. And you have a.
Oh no, I don't. It's fine. You guys get it. Okay, so
You point this pointer, so you can just.
yeah, it's okay. It's no problem. I'll just keep walking back and forth. So I'm going to do a quick overview of kind of our technology, what sets us apart, why one would go with the G4 sequencer over other sequencers on the market. And then for those of you who would like to submit libraries to Yuri's lab for sequencing or are interested in using the G4 for sequencing for yourselves, I'm going to talk through a little bit about how to adapt your current libraries, mostly Illumina libraries, to run on the G4 and kind of different ways to sequence. And then we'll wrap up, what I didn't put on my agenda is that Jonathan's going to talk about our newest instrument that is going to be launching next year, which is our G4X, which is going to incorporate a spatial element. And so Jonathan will talk about that, and then we'll just do a quick summary and have time for questions. But this is, I say it's informal, this feels very formal, but it's informal. Raise your hand, ask questions at any point. Okay, so just a little bit of a technical overview. So the G4, we developed the G4, our company developed the G4 in order to address 4 needs that we felt like the market was missing for the next generation sequencing market. The first one was flexibility. So for most Illumina sequencers, they rely on very big flow cells, so they want to maximize the output of a flow cell, which is great if you have a lot of samples or if your samples need a lot of reads. But there's a number of labs that only want to do a quick research project. They only need a few 100 million reads, maybe just a few million reads. And so what we wanted to do was have the same throughput, the same output, which is going to be around 1.2 to 1.6 billion reads per run. But we wanted to break that down into smaller pieces so that if you wanted to only do 400 million reads at once, you could. So the way that we address that is by having the ability to run up to 4 flow cells at once. So you can run a 1 flow cell or 4 flow cells, which you can see here. These are our flow cells. And then within each of those 4 flow cells, there's 4 independent lanes. So you could hypothetically have 16 independent experiments running at once, or you can have the smallest kind of unit be 4 independent experiments at once. You can also do 1 very large experiment and just run the same library across all 16 lanes and get that maximum throughput for your library. So it just gives you the added flexibility, but still the same throughput of like a NextSeq, a mid-level NextSeq run. The other thing we wanted to address is the speed. So all of our sequencing runs, our longest being the 2x300 with some indexes in there, are going to complete within 24 hours. Add on maybe an hour or so for analysis, which is also done on the instrument. So you could hypothetically, and what we do at Singular, is we start a run every single day on our system. So every day around the same time, we're starting a run. And because of our throughput and our speed, we have the most powerful benchtop sequencer in that we're able to output the most data at a time in a single day. And finally, because we have 4-color chemistry and we have what we call pattern flow cells, which I can get into the details of that if anyone is interested. But because of those 2 things, we have industry standard accuracy without the need for Phi-X to be added in, which I'll get to in some future slides. Okay, so let's talk about flexibility in a little bit more detail. So as I mentioned, it allows for the user to run up to 4 independent flow cells at a time, and each of those flow cells has 4 independent lanes. So I don't know if you guys are familiar with submitting libraries in for sequencing, but oftentimes you may be waiting like a month or 2 months at a time because you have to wait for enough libraries to fill a very big flow cell, and you have to make sure that your library has different sample indexes than the next library so that you guys don't get mixed up. With our flow cells, because everything is independent, you can have your own lane, and you can run the same sample indexes in different lanes, and those will get demultiplexed separately. Does everybody understand what that means? Okay. And so because of that, you can have a much faster turnaround time when you're submitting samples to a core, because they can get your flow cell done in 1 day, right? You don't have to wait to get 25 billion reads for a NovaSeq. You can just run your 1 flow cell that has 400 million reads that has maybe only your samples on it. Or if you only need 1 lane, I'm not sure how Yuri's going to do it, but if you only need 1 lane, maybe you just buy 1 lane, or you combine with other labs and you get 1 lane and 3 other labs get 1 lane, and then you guys can run your flow cell and then get that data back within 24 hours or a week or whatever the time is without having to wait to pool samples. Also, if somebody else messes up their libraries and you then have to pull it, let's say on an Illumina library, and they mess up the whole run and you don't get any data back, that won't happen here. You never have to mix your libraries with anybody else's library, so your samples will get sequenced independently and hopefully successfully, making sure that you made your libraries correctly. Any questions on that? Yeah.
Will you talk about how to convert an existing Illumina library to run on the singular?
Yeah. I'm going to talk about that probably the most. So yes, and I have some good slides on it. I've just here, this is just a graphical representation of how you can split up a flow cell if you wanted to. Again, with the independent lanes and independent flow cells. So let's talk about what kind of flow cells we provide. So we have two size flow cells. We have the F2 flow cell, which is our lower output flow cell, and our F3 flow cell, which is our higher output. Each of those can go up to 300 cycles, but we have a 100 cycle kit, 200 cycle, and a 300 cycle. And each of those kits has an extra 50 cycles. So the 100 cycle kit's perfect for like a 10x library with 10 base pair indexes. And then you can kind of mix and match with some of our other libraries depending on your application, RNA-seq, whole genome, whatever you're doing. For our F2 flow cell, you're gonna get around 200 million reads per flow cell. So that's about like 50 million reads per lane. And then for our F3 flow cell, you're gonna have 400 million reads per flow cell. So that's 100 million reads per lane. And then you can multiplex within that lane. So if your libraries only need 10 million reads, you can have 5 to 10 samples per lane, depending on what your application is. We mention a lot of this in reads, but it's actually clusters. So when I say 400 million reads, I mean you get 400 million read ones and 400 million read twos. So that's 400 million paired reads. I know Illumina they kind of do a weird thing with their single end and paired end reads, but this is 400 million clusters, meaning 400 million paired reads. Any questions on that? Okay. Finally, our quality, you're going to get greater than 85% bases over Q30 with all of our reads, all of our kits. So as I mentioned, we have 4-color chemistry. That just means that each of our nucleotides has their own independent fluorescence. We think that this adds to our accuracy. With Illumina, some of their instruments have 4-color chemistry, others have 2-color chemistry or 1-color chemistry, where they're inferring a base based on a combination of fluorescence. You can oftentimes get errors associated with Gs or long Gs because that's the nucleotide that has no color on the Illumina instrument. Again, Illumina is great. They're the pioneers in this system, but I'm just giving you the comparison as to why somebody would choose the G4 over an Illumina instrument. Finally, our runtime, which I already mentioned. So we're going to complete all of our runs in less than 24 hours. On other instruments, you're going to see that be doubled, 36, maybe 48 hours, including analysis. Finally, we do our base calling and analysis all on board the instrument. So you input your library, and what you get out are going to be your demultiplexed FASTQ files. And so those FASTQ files should be able to fit seamlessly into your downstream bioinformatics solutions that you have already. Our data looks very similar to Illumina data, so most of our customers don't need to do too much tweaking to their bioinformatics pipeline. They can just input our FASTQ files into what they're already using, and they get very similar data out. So this just shows a general overview of how we wanted the G4 to be a plug and play solution. We didn't want you to have to change your existing library prep. We didn't want you to have to change your analysis on the back end. We just wanted you to have that sequencing solution that fits into your existing kind of ecosystem that you're running. So now I'm going to get into that library prep. So we've partnered with over, I want to say 18 different library prep providers, and we have tech notes with them. Some of them we have protocols that have been updated to be singular specific. Others, we've just run their data, run their libraries on our sequencer and shown that the data looks excellent. And all of that can be found on our website. And I'll have a link to that at the end of the PowerPoint. There's two ways that you can get your libraries ready for the G4 sequencer. You can either do like a de novo library prep, where you're introducing our adapters and PCR primers during the library prep, so that You don't have to do any conversion. You just make a library that's singular specific already. Some things to note. Our sequencing primers, which we label here as SP1 and SP2, they're going to be the same sequence as the Illumina TrueSeq primers. So you don't need to have any kind of custom primer situation for the G4. The difference comes in our flow cell binding sites. So you guys may be familiar with the Illumina flow cell binding sites, which are P5 and P7. We don't use those. We use our singular specific flow cell binding sites, which we call S1 and S2. So the way that you would get your library to be singular compatible is just by having, instead of P5, P7, the S1 and S2 sequences. So what that means is that for most, not all, but most library preps on the market, you would just change the final PCR amplification step. So instead of amplifying with your index primers that have the P5 and P7 at the end, you would just grab your singular primer tube and amplify your libraries up with the singular adapters. The landing site for that primer is going to be the same. It's that SP1 and SP2, which is the TrueSeq sequencing primers. But instead of extending on the index in P5, P7, it'll extend your index in S1 and S2. So that's the easiest way to get your libraries to be singular compatible without having to do a conversion after the fact. Are there any questions on this? This is like a very general overview. Different library preps are going to be a little different. Yeah.
Another question.
Of course.
If I already have a multiplex sample of many Illumina libraries already barcoded, just want to do that one adapter extension protocol?
So for that case, we have our compatibility kit, which is basically our conversion kit. That will bind off of the P5 and the P7 and then add on our S1 and S2 sequences. In that case, it'll retain your indexing structure. So if you already have a pool of libraries, it'll maintain those indexes within that pool, but just add on our S1 and S2. So that cuts down, one, on the number of times you have to do a conversion. You can just convert your entire pool, but it also maintains your index structure and index kind of strategy.
Okay.
Yeah. We sell this kit with the primers, so the S1 with the kind of P5 overhang and the S2 with the P7 overhang as a kit. So you just order that, convert your library pool. This conversion is between 4 and 7 cycles of PCR. So it usually takes in the lab about 20 minutes, plus a bead purification. So you're looking at maybe 45 minutes to an hour to go from your Illumina library to your singular compatible ready to sequence library.
Do you lose any bases from your sequencing?
No, you do not lose any bases from your library if you do that. Yeah. So because we are, so what I haven't gotten into, it's actually on the next slide, but what I haven't gotten into is how we actually do our sequencing. So just as a comparison, for Illumina, does this have at least a pointer? No. Okay, let me get on this side. Okay, so this side is going to show you kind of the Illumina read structure. So they bind the SP1 or the TrueSeq sequencing primer here and sequence your index in that direction. And then they're gonna, well, first they start with their insert. So they bind the SP1, they sequence your insert, then they bind the index from the inside and sequence out. And then they bind out here and sequence your index 2 from the outside in, and then they come back on the second strand, lay down a primer, and then sequence their insert, okay? We do our strategy a little bit differently in that we sequence our index first, our index 1 from the outside in, so like our S1 sequence our index. Then we lay down a primer on the SP1, our sequencing primer 1 site, sequence our insert. Then we come to the second strand, lay down a primer on S2, sequence our index, and then lay down a primer on SP2 and sequence our second insert. So because of that, I'm just gonna go back one. Because of that, we don't lose any actual information when we're sequencing a converted library. We're laying down our primers in the exact same place. We do provide kind of this hybrid library that will, hybrid primer that will bind this little piece of P5 as well, so you're only sequencing your index. Similarly, on the other side, it's a hybrid primer that binds this little piece of P7, so you're only sequencing your index. And then your insert sequencing primers remain the same, so we sequence those as per usual. Lay down your sequencing primer 1, sequencing primer 2 and sequence your insert from both sides, if you're doing paired end sequencing. Did that answer your question? Okay. Any other questions on this? Or how we do our sequencing strategy? Okay. So because of how, because we changed the way that we sequence our indexes mainly, the way that you make your sample sheet for the G4 is going to be different than you would make your sample sheet for for Illumina, in that our index 1 is Illumina's index 2 and our index 2 is Illumina index 1. If you are going to create a singular library, I have left my card up here so that you can email me if you need help with adapting your libraries, creating a sample sheet, how would you work your current library prep to make it so that you don't need to convert. All of those questions I love answering. They're so much fun. Happy to answer them to help you get going. But this is just a quick overview just so that you guys get familiar with the technology. Any questions on any of that? No? Okay. As I mentioned, you have the option to do your de novo library prep. We do sell, our library prep adapter and our Singular Genomics unique dual index primers if you wanted to create your own kind of libraries de novo using off-the-shelf library preps or your own kind of homebrew library prep. We sell a non-index PCR-free library preps for those who use that kind of unique library prep method for usually whole genome. And then we also have non-index PCR primers. Finally, for those who want to do those conversions, we do sell that G4 library compatibility kit. It comes in with 24 reactions possible for that. And that's not 24 samples, but that's 24 reactions. So if you have a pool, that would be considered one reaction. So you can do a lot of libraries with one compatibility kit. Any questions on our products? Yeah.
I saw on the list you had some companies, but Zymo wasn't one of them.
Mm-hmm. Yeah, it is not. I feel like we have a customer who uses the Zymo kit. Again,
we do a lot of RNA-seq with Zymo.
Okay.
So we would need to be able to convert it.
For sure. You can either convert it if you wanted to, or I'm pretty sure we have one customer who incorporates our primers into that last step of PCR for the Zymo kit so that there's no need to convert. It's already in that singular format. But I'm sure that they have done both. So I think they started off converting it, and then once they felt comfortable with it, they ordered the singular primers and then just started making the libraries de novo. Right. So yeah, there's a number of library preps that are not listed that we have worked with, just not officially. Yeah.
How does it work with the low complexity samples?
Yes, good question. So let me.
Before you can spread your sample with other researchers or other experiments.
Yes. Great question. Let me fast forward to this slide. So one thing about the G4 sequencer is because we use 4-color chemistry and we have what is called a pattern flow cell, meaning ideally one DNA molecule will have its own like nanowell where it's getting amplified into a cluster. We're able to sequence low complexity libraries without the need for Phi-X added or without the need of other libraries creating complexity on the flow cell. So on the left, my right, you can see that we have 4 different it's the same library, but with 4 different percentages of Phi-X added. So we have 0% Phi-X at the bottom and 50% Phi-X added at the top. This is a methyl-seq library, so it has an overrepresentation of 3 of the 4 nucleotides, causing it to be somewhat low complexity. You can see that even with 0% Phi-X, we got 92% of our bases over Q30. That doesn't change when we add in 50% Phi-X to increase the complexity. So you can add in just the baseline, either 0% Phi-X, which I don't recommend. I always like to have a little bit of an internal control. We recommend 2% Phi-X for all of our runs. So we have customers who do 1 amplicon library, so that means 1 section of the genome, so everything looks exactly the same on the flow cell. They still add in 2% Phi-X and still get 400 million reads, Q30 scores above 90%. So you don't need to have complexity in order to have successful sequencing on the G4 platform. Any other questions on that? On this side, this is showing you that one library, this was with an external partner, they had an A nucleotide at the same position for every single DNA molecule in the flow cell. With Illumina, you can see that quality dropped down to Q10, but with the G4, it stayed high and remained high throughout the rest of the run. So again, just another example that you don't need that complexity to have successful sequencing on the G4. Any other questions on that? Okay, I just have a couple more slides before we get Jonathan to come up and talk about spatial, which I know we're all excited about. I just wanted to quickly show that the data coming off the G4 is comparable to like the Illumina NextSeq instrument. This data specifically is for RNA-seq data. The first graph is showing you 2 replicates on the G4, just showing you that they're highly correlated, so you get reproducible data on the G4. The second graph is comparing Illumina on the y-axis to G4 on the x-axis, again showing you that we get the same transcript counts whether you're sequencing on an Illumina instrument or the G4. Finally, or not finally, but in the third graph, we're showing G4 detected genes versus those detected on the NextSeq, again, quite comparable. And then finally, the coverage across the sequencing for both the G4 and the NextSeq, again, comparable. So if you're used to sequencing on an Illumina system, feel comfortable transitioning to a G4 without sacrificing your data, but with the added benefit of doing smaller run sizes at cheaper cost and faster turnaround time. And this is single cell RNA-seq. Again, very similar results on both the G4 and the NextSeq in terms of cell sorting, as well as the gene expression that was detected for those single cells. On our website, we have a number of tech notes. We have a lot of library prep protocols for the different library preps. We have a general library prep protocol if you wanted to do de novo. We have our conversion protocol on there. We have videos on how to load our system. All of that can be found at singulargenomics.com/resources. And you can find all of those videos, application notes, posters, publications. We have FAQs, all of that stuff. Demo data, FASTQ files if you're a bioinformatician and want to see how that fits into your pipeline. Again, feel free to give me an email or call if you have any specific questions related to your application. Any questions on the G4 sequencing side of things before Jonathan gets going on the spatial side of things? Awesome. Thank you, guys. Oh, yeah, mic.
And just go through.
Yeah, I turned it off, so it's not bad.
Yeah, best. Need my readers. Yeah. Good. Hello. All right. Thank you, everybody. Thank you, Jordan. Jordan is going to be a fantastic asset for you if there's single cell RNA-seq libraries. Again, the really key to the G4 is that ability to scale down and scale up. So if you have just one library you want to run, there's a scale for that. If you have a bunch of them, we can kind of scale up. And again, that date in a day, really fast turnaround times. Just reach out to Yuri, some really great pricing as well on all those F2 and F3 kits. But the G4 itself, pretty interesting, right? When we take the cover off, we've got NVIDIA GPUs, really fast processing. We've got fluidics that have these 4 flow cells with 16 lanes. We've got ultra high speed imaging. We've got precision heatings and fine-tuned microfluidics. We're thinking to ourselves, what could this really, turn into? And it's actually something that we're excited to be launching next year, which is the G4X. It's our in situ multi-omic analysis for FFP samples at subcellular resolution. So the base of the G4 had basically everything we needed, minus the reagents and some different things I'm going to show you, to become a spatial instrument and be able to look at and profile tissue across multiple flow cells, which now is going to equate not just to flow cells and libraries, but image area and how many samples you guys can look at. So we're really excited for this to be coming out when we think about this, thinking about integrated multi-omic data sets. So there's 4 things that the G4X will be able to do. Transcriptomics, proteomics, and fluorescent H&E will be available at the launch, and they're actually available now as part of our technology access services. And then DirectSeq is something that will be coming out a little bit later after the launch. DirectSeq, a really, really cool application where we're looking at TCR, BCR profiling, looking at cancer mutations, looking at CRISPR gene editing, all within your tissue. So really cool. I'm going to go into this in a little bit. But the first thing is transcriptomics. We're going to have barcode-free profiling of hundreds of genes. We'll have proteomics as well. And then a fluorescent H&E, which is just to get your morphology. It's not going to replace your standard H&E, but it's kind of nice within one section of tissue to be able to get these modalities. So how does the G4X work? The workflow is pretty basic. So it's prepare, right? We've got a tissue transfer tool that we use to streamline sample preparation and get your samples onto those flow cells. There's also a custom panel designer and certain sets of panels that we will offer, which I'll go through a little bit later. Once you have your samples prepared, basically it's the same thing as kind of putting your library on the G4. Now you're going to be putting your flow cell with your tissues onto the G4X, where we'll do this high-speed imaging and analysis of those tissues and those targeted transcriptomics. And then last but not least, which is obviously the biggest bottleneck for everybody, is that analysis, right? Getting to do your analysis. There are certain data files that you'll get. You'll get a CSV with your your transcript counts, your localizations. You're going to get TIFF files of all your protein and your segmentation data. And then you can put this into either our viewer or third-party analysis to do some of your spatial analysis downstream. When we look at the workflow, I just want to kind of dive a little bit deeper into this kind of tissue transfer. Quick raise of hands. Has anyone done Visium, or has anybody done kind of spatial work? All right, we got 1, 2, 3, 4. So I used to work at 10X and I launched Visium, and I still remember trying to scoop that FFPE section out of the water bath onto the slide. Tissue prep for anything spatial is going to be pretty much some of the hardest things that you're going to do in order to get to the downstream, which is that analysis. So what Singular's done is kind of come up with this little tissue transfer tool. And what you'll do is you're going to slice your tissue sections onto a hydrogel slab. You have a little puncher. It's going to be in 2 sizes, a 4x4 or a 10x10. So you actually punch out your tissue, you place it onto this holder, and then that's how you'll line up all your tissues. You create a little flow cell sandwich, and then you're going to get your tissue onto your flow cell. So think of this as your standard FFPE section 10x10 millimeter, or you can take these 4x4 millimeter punches. And after you do this tissue transfer with that kind of sandwich, you align your tissues onto these flow cells so that they look either like this with the 10 millimeter samples or like this with the 4 millimeter samples. It's as easy as possible. Now, I'm going to say it is not a trivial process. It still takes training. It still takes work. Handling tissue is something that is just, if you're used to it, it's cool. But if it's something new to you, this will take some training. But this is as innovative a way as I've seen to be able to get that many sections of tissue onto those flow cells, which then we're going to start to do our analysis on. So once the tissues are on the flow cell, there is some standard molecular biology. Sure.
Could you just say that section that you're talking about? So that's a regular microscope slide that you're punching off of. What are you punching?
Yeah. So you're actually you're slicing. You're going to section your tissue onto a hydrogel slab, and then you're punching through that hydrogel slab to get the tissue with that little bit of the slab. Then you invert that cutter, place it on here, and then your tissue's kind of at the top so that it makes that little sandwich that we can then get your tissues off.
Can you talk a little bit more about how you process that tissue on the slab? Where it's coming from?
Yeah, it's coming from a standard FFPE block, and then it's just using a microtome, taking your little ribbon, and then sectioning it onto your hydrogel slab, either in a water bath.
So we provide the slab we provide, it would come with the kit. It's like on a holder. So it looks kind of like this flow cell, but it's just a slab of hydrogel it's on a holder, so it looks similar to your process, except it's not like a slide that you would slide under the water. Instead, you would slide your hydrogel under the water, capture your tissue onto that hydrogel. So it looks like your tissue is on a slide, but it's just a hydrogel. That you can keep for up to a week on that hydrogel, and then you can punch it out if you want to. But you just, like Jonathan said, punch it out from that hydrogel. But it's something we would provide to you. You don't need to like make it or find it somewhere. Yeah.
So there are individual bars or boards that you move?
They're cutters. It's like a cookie cutter.
Yeah, it's like a little cookie cutter. So a little metal piece that will slide through the tissue, and then you take that off and you place it on the holder. You put another cutter in. You make your next one. You slice that. Put it on the holder.
If you already have like a TMA and you want
Yeah.
to image the whole TMA, or you have a really skilled histotech who's like slicing's no problem, can you skip this and just put your tissue without the whole punching part of it?
Yeah, we haven't tried that. The TMA is going to be that 4x4 size. We haven't tried that. This is kind of our process. That's not to say it couldn't happen, but I would lean towards saying no right now because of the way it has to be aligned on that flow cell. So you can't really just, it's not like a permafrost slide that you have your microtome and you're kind of like slicing right onto it. This is actually different than kind of this setup, right? It's kind of separate, right? So you're slicing onto your hydrogel, which is here, you're cutting, and then you're transferring. So it's not like you can just slice onto this kind of flow cell. No matter how great the tech is, I just don't see how that happens without this kind of tool.
So is that multiple independent samples from independent tissue slices potentially?
Yeah, so it could be serial sections, or it can be one block, two. Again, for here, most of our customers are running like 10 different samples, right? So 10 different blocks, one section each. But the power of the throughput, which I'm going to show you in a second, means that we can start doing maybe more serial sections and starting to see deeper into that block and do some kind of 3D reconstruction and things like that. So it's going to be up to your experimental design. We can talk afterwards if you want to get a little bit deeper into kind of that as well, because I see you're still kind of going.
But you're essentially building an array onto the slide directly.
Yes, exactly. You can think of it from that perspective. So then the molecular biology is you're going to do your antigen retrieval. You do your deparaffinatation, you de-cross-linking. You'll do 2 different proteomic labeling along with your transcriptomic labeling, and then you're now ready for your in situ sequencing. So there is about a day and a half of molecular biology that happens before it gets onto that. So you do your tissue sectioning, then you're going to do your molecular biology, then you're going to put it onto the imager. And again, this is what's fantastic, thinking about that G4 where we talked about the scalability, right? So there's flexibility, first of all, in configurations. You can do the 4x4 configuration, which does look like a TMA. You can do 32 different samples on this. There's also our 10x10 samples where we can do 10 10x10 samples. And then there's a smaller flow cell that'll fit 4 10x10 samples. So you have some kind of flexibility in the amount of samples that you want to run, as well as kind of that imaging area, right? So we have 10 centimeters squared versus here, which would be 4 centimeters squared. So you have your flexibility in the sample size and you have your flexibility in the imaging area. And again, all this equates to is that ability, depending on what you have at hand at that time, just like your library. So if I have one single cell library, I can go to the G4 and run it tomorrow. If I have a few sections, maybe I want to use the small flow cell. If I have a large kind of study, maybe I'm going to do the 32 samples and do that one flow cell. Maybe I have more than that. So again, you can kind of scale up your experiments or scale them down depending on what you have at that time. And you can scale anywhere from 4 centimeters squared up to 40 centimeters squared per run. And when we think about what 40 centimeters squared means, this is the imaging area of the G4X spatial sequencer. This is the Cosmics, this is the Xenium, and this is the MERScope. So this is how much imaging area the other systems can do currently. This is how much the G4X will be able to do utilizing those 4 flow cells. That doesn't mean you have to run 4 flow cells. You can run 1 if you want, you can run 2, but there is this expansive imaging area, which I think is going to speak to more high throughput type of studies, where it could be more sections instead of more, or it could be more samples. It could be a little bit of a mix of both. So simply, how does it work? The transcript counting, we put in a padlock barcode. We'll do your rolling circle amplification. And then using the sequencing technology, same as the G4, we'll come in here with a primer, and we're just going to start reading that transcript. So we're actually going to be sequencing ATTs, GCs, instead of some of these barcode or these fluorescent hybridization technologies that are out there. I'm sorry. For DirectSeq, which I was telling you about, DirectSeq is about looking at unknown regions of our mRNA. So you have a known region, a cancer mutation, right? We know kind of the anchors. What we'll do then is we'll take a reverse complement of this unknown region. We will still do the rolling circle amplification. And then again, we're just going to kind of sequence that out. So for TCR, BCR, we can get that unknown region, cancer mutation hotspots, and CRISPR editings. This is going to be a really cool application, I said later. And then from a protein perspective, it's just your ab oligo. So we have an ab oligo, we're gonna put our probe on it, and then instead of reading different cycles, we're just gonna read one cycle at a time and do 4 colors. So each of your proteins are gonna be A, T, G, or C. We'll do 4, cycle through, do another 4, cycle through, do another 4. And currently we're doing 12 proteins as part of our panel, though we see this getting up to over 20 by the time that we do launch the system. So slightly different aspect. This is actually directly sequencing those probes versus bringing in these fluorescently hybridized barcodes that then give the single and look at it. So slightly different way than Cosmic, Xenium, and Merscope, but still getting that subcellular in situ resolution. So from an analysis perspective, there's a web visualization tool out there that you can kind of look at. You can look at all your genes. We can zoom in and zoom out of different tissue sections just to kind of check out that resolution of not only your transcript, but your proteins. And then if we want to start looking at, some data, this is just tonsil. So this is a 294-plex. When we look at transcript protein, then we can start doing our cell typing, then we can start looking at community-associated cell typing. And then again, all of the different type of analysis that goes into kind of a spatial experiment. How many unique transcripts per cell? What's my average genes per cell? What's the false discovery rate? Everything that you're seeing kind of in this spatial world are the things that you're able to get out by the basic formats that come off of the G4X. And again, of course, at scale, where you're going to see kind of your transcripts, your total number of cells. So again, think of these as spatial single cell experiments where you can then kind of look at all of your cells across this tissue. How many transcripts are there, your median transcripts, your median genes, all the kind of data that we get out of the G4X. And then just to kind of show you something a little bit different, this is lung in healthy versus disease.
Do you have an idea about detection sensitivity?
Sensitivity from a perspective,
like if there are like 10 transcripts in a cell, how many of them do you detect?
Yeah, so we don't have that exact data yet, those data sets yet. This is something we're trying to build out through not only through our technology access program, but obviously through our own data sets. But more to come, probably the next 2 months is when we're going to be releasing our first public data set that will actually have that exact sensitivity data from there. Not sure how that's going to relate compared to a Xenium or Merscope, but again, this is definitely one of the questions that we get is kind of where are you at from a sensitivity perspective.
And then maybe a related question, like I think the poster I looked at, you have like 150 probes for genes. Can you go up?
Yeah, so
how far?
Yeah, so
how does that relate to detection?
You're right. So that actually adds a lot to detection. So currently we're building out our panels with 12 probes with the ability to go up to 20 or even 30. And it's just about iterating them, right? In order to get them kind of where we need to see that sensitivity. So the first kind of data sets I said are going to be coming out in about a month or two. Those are around 12 probes per gene, but you can definitely see that the next iterations will probably have more increased sensitivity as we do increase those number of probes. And again, this is just looking at lung tissue versus disease tissue. Obviously, you can see different lung anatomy and kind of the gene structure that's going on there. And then when you start to look at disease, you can just see the different types of genes that are going to be coming in. These are all the kind of fancy spatial pictures that you will see, but there is a lot of great depth to some of this data when you start looking at it. Just to kind of end, what the G4X really gives you is that scale, right? So scaling a spatial biology program. Today, you're really limited by throughput and price. If you're going to do a Xenium run or even a Visium run, it might be 2-4 sections at the most. It can be quite expensive. What could you do if you could do more sections, right? So more samples. Or if we take that, if we do more sections from those samples, this is really where we get to the power of what the G4X can give you. More sections across more samples, increased throughput at a reduced price. So currently, we are offering G4X services. So if it's something you would like to test, this is something we would love to talk with you about. It starts with initial consultation. We'll process the samples, provide you the data, and actually you will get a free flow cell. So you'll get 10 sections that we can do as a pilot for you guys at no charge if there is a project behind it. Again, something I'd be happy to discuss with you. And from that perspective, we're looking at lung, kidney, breast, colon. Those are kind of the 4 sample types that we're going to start with. We have an immuno-oncology panel, and then we have these specific panels that are built on 200 genes from an immuno-oncology perspective, with 100 plus 100 genes that are specific to lung, kidney, breast, and colon. So if you're working in any of these tissues and you're interested in potentially seeing some data on the G4X, we would love to kind of chat with you on that. There's also our protein proteomics panel. So we validated 15 markers on the system. There's 12 currently in the immuno-oncology panel that comes with that transcripts and proteins, but something that, again, we're looking to continue to build out. And just in closing, it's fantastic, that we have the G4 here with Yuri, but it is 2 technologies in 1 instrument. So you can do all of your next-gen sequencing on it, and then when the upgrade is available, you can do the spatial stuff. So one day, if you have spatial, great. The next day, maybe you want to run your Visium libraries or your single-cell libraries for those spatial. So within 2 days, you can actually get spatial and your library and your single-cell or your next-generation sequencing library data from that. Really, really excited about the potential of this. And again, not only from the sequencing perspective, please, as I said, scaling down, getting those 10x libraries, those RNA-seqs, 200 million reads up to 1.6 billion reads. We'd love to start working with that. But again, if you also have a spatial project in mind, we would love to work with you as well on that. Because it'd just be great to be able to have data so that we can then go back to Yuri or the team here and say, hey, you know what, this stuff really looks great. This is something that we need to get invested in. And we would love to work with you guys on that. So with that, I am finished, but I'll be hanging around for a while. I know Jordan can hang out for a little bit. She's got to get to a plane, but we're here to answer any questions that you guys might have. But thank you for coming. There's still a couple salads left. There's some cool Panera bags. I love them for camping, if you want to grab those. But thank you guys, appreciate your time. Yes.
So talking about upgrades, your cells are the same. So the cells are going to be the same.
The flow cells?
Yeah, the flow cells.
No, the flow cells are actually differently engineered. So they're for sequencing, you have those pattern flow cells that have those like tiny little nanowells in it. It's a totally different. It's similar in the fact that you can put 4 together, and they have that. One of them has the 4 lanes on it for the 4x4, but it is a totally different flow cell. It's actually different to allow microfluidics and different fluidics to kind of flow through the system.
But you can load them. You can load them in the same positions, right? So it's a similar flow cell anatomy that it can be loaded in the same place, but it's not the same exact flow cell. There's a spatial flow cell and an NGS flow cell, but they fit in the same.
So the instrument is going to be the same.
The instrument's the same.
So the upgrade is the same.
We have
the upgrade is mostly
3D data sets, big data sets. We tested spatial genomics, sequence. We have our own virtual platform.
Nice.
Add more tissues. So one thing, my question I have. Yes. Yeah. So one question I have is actually is what is the success rate? Like you can have multiple samples.
Yes. So
One go, which is phenomenal.
Exactly. And that's one thing that I work on. It's like what happens when you have a failure. So right now we're
DC
looking.
microlithing.
So
we've
we've seen a little bit of breast, a little delamination.
nothing
Nothing too crazy. And again, I think our internal retest rate is around 20.